Next, we're going to quickly skim through all these points. Cytokine profiles are cross-regulated. The critical cytokine produced by Th1 and Th2 subsets have two characteristic effects on subset development. First, they promote the growth of the subset that produced them. Second, they inhibit the development and activity of the opposite subset, an effect known as cross-regulation. For instance, IFN gamma secreted by the Th1 subset preferentially inhibits proliferation of the Th2 subset, and IL-4 and IL-10 secreted by the Th2 subset downregulate secretion of IL-12, one of the critical cytokines for Th1 differentiation by both macrophages and dendritic cells. Similarly, the cytokines have opposing effects on target cells other than Th subsets. IFN gamma secreted by the Th1 subset produce IgG2A production by B cells but inhibits IgG1 and IgE production. On the other hand, IL-4 secreted by the Th2 subset promote production of IgG1 and IgE and suppress production of IgG2A. The phenomenon of cross-regulation explains the observation that there is often an inverse relationship between antibody production and cell-mediated immunity. That is, when antibody production is high, cell-mediated immunity is low, vice versa. Furthermore, recent research has shown that IL-4 and IFN gamma make members of the T cell subset that release them less responsive to the cytokine that direct differentiation of the other T cell subset. Thus, IL-4 enhance Th2 cell development by making Th cells less susceptible to the cytokine signals that cause these cells to enter a differentiation pathway that would lead to Th1 development. On the other hand, as explained below, IFN gamma upregulates the expression of a key regulatory molecule that favors the differentiation and activity of Th1 cell. Recent work has given insight into the molecular basis for the cytokine-mediated cross-regulation by which one subset promotes its own expansion and development while inhibiting the development of the opposite subset. Two transcription factors, Tbet and GATA3, are key elements in determining subset commitment and cross-regulation. The expression of Tbet drives cells to differentiate into Th1 cells and suppresses their differentiation along the Th2 pathway. Expression of GATA3 does the opposite, promoting the cross-regulation of Th1 cells by IL-10. Development of naive T cells into Th2 cells while suppressing their differentiation into Th1 cells. The expression of Tbet versus GATA3 is determined by the cytokines IFN gamma and IL-4. In the presence of IFN gamma, T cells upregulate the expression of Tbet and downregulate GATA3. This IFN gamma receptor STAT1 dependent process shifts the cytokine profile to the production of IFN gamma, the signature cytokine of Th1 cells and other cytokines typical of the Th1 cell. On the other hand, in a process that involves the IL-4 receptor and STAT6, IL-4 induces the cell to produce IL-4 and other Th2 cytokines. Further study has revealed that the upregulation of Tbet represses the expression of GATA3. Similarly, expression of GATA3 downregulates Tbet. Consequently, cytokine signals that induce one of these transcription factors set in motion a chain of events that repress the other. In the intracellular level, the differentiation of a T cell along the Th1 pathway prevents its development of Th2 characteristics and vice versa. The cross-regulation of Th1 cells by IL-10 secreted from Th2 cells is not a direct inhibition of the Th1 cells. Instead, IL-10 acts on monocytes and macrophages, interfering with their ability to activate the Th1 subset. This interference is thought to result from the demonstrated ability of IL-10 to downregulate the expression of class II MHC molecules on these antigen-presenting cells. IL-10 has other potent immunosuppressant effects on the monocyte macrophage lineage, such as suppressing the production of nitric oxide and other bacterial metabolites involved in the destruction of pathogens, and also suppressing the production of various inflammatory mediators. These suppressive effects on the macrophage serve to further diminish the biologic consequences of Th1 activation. The Th1 Th2 balance determines disease outcomes. The progression of some diseases may depend on the balance between the Th1 and Th2 subsets. In humans, a well-studied example of this phenomenon is leprosy, which is caused by Mycobacterium leprae, an intracellular pathogen that can survive within the phagosomes of macrophages. So, leprosy is not a single clinical entity, rather, the disease presents a spectrum of clinical responses with two major forms of disease, tuberculoid and lepromatous, at each end of the spectrum. In tuberculoid leprosy, a cell-mediated immune response forms granulomas, resulting in the destruction of most of the mycobacteria, so that only a few organisms remain in the tissues. Although skin and peripheral nerves are damaged, tuberculoid leprosy progresses slowly, and patients usually survive. In lepromatous leprosy, the cell-mediated response is depressed, and instead humoral antibodies are formed, sometimes resulting in hypergamma-globulinemia. The mycobacteria are widely disseminated in macrophages, often reaching numbers as high as 10 raised to 10 per gram of tissue. Lepromatous leprosy progresses into disseminated infection of the bone and cartilage with extensive nerve damage. The development of lepromatous or tuberculoid leprosy depends on the balance of Th1 and Th2 cells. In tuberculoid leprosy, the immune response is characterized by Th1 type response, that later type hypersensitivity, and cytokine profile consisting of high levels of IL-2, IFN gamma, and IFN beta. In lepromatous leprosy, there is a Th2 type immune response with high levels of IL-4, IL-5, and IL-10. This cytokine profile explains the diminished cell-mediated immunity and increased production of serum antibody in lepromatous leprosy. There is also evidence for change in Th subset activity in AIDS. Early in the disease, Th1 activity is high, but in disease AIDS progress, some workers have suggested there may be a shift from a Th1-like to Th2-like response. In addition, some pathogens may influence the activity of the Th subsets. The Epstein-Barr virus, for instance, provides VIL-10, which has IL-10-like activity, and like cellular IL-10, tends to suppress Th1 activity by cross-regulation. Some researchers have speculated that VIL-10 may reduce the cell-mediated response to an Epstein-Barr virus, thus conferring a survival advantage on the virus. And cytokine-related diseases. So this portion is, something related to the clinical application. Actually, this is not involved in the syllabus, but still I'm gonna read this. Defects in the complex regulatory networks governing the expression of cytokines and cytokine receptors have been implicated in a number of diseases. This section describes several diseases resulting from overexpression or underexpression of cytokines or cytokine receptors. Bacterial septic shock is common and potentially lethal. The role of cytokine overproduction in pathogenesis can be illustrated by bacterial septic shock. This condition may develop a few hours after infection by certain gram-negative bacteria, including E. coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Enterobacter aerogenes, and Neisseria meningitidis. The symptoms of bacterial septic shock, which is often fatal, include a drop in blood pressure, fever, diarrhea, and widespread blood clotting in various organs. This condition afflicts about 500,000 Americans annually and causes more than 70,000 deaths. The annual cost for treating bacterial septic shock is estimated to be in excess of $5 billion. So bacterial septic shock apparently develops because bacterial cell wall endotoxins stimulate macrophages to overproduce IL-1 and TNF-alpha to levels that cause septic shock. In one study, for example, higher levels of TNF-alpha were found in patients who died of meningitis than in those who recovered. Furthermore, a condition resembling bacterial septic shock can be produced by injecting mice with recombinant TNF-alpha in the absence of gram-negative bacteria infection. Several studies offer some hope that neutralization of TNF-alpha or IL-1 activity with monoclonal antibodies or antagonists may prevent this fatal shock from developing in these bacterial infections. In one study, monoclonal antibody to TNF-alpha protected animals from endotoxin-induced shock. Another study has shown that injection of recombinant IL-1 receptor antagonist, which prevents binding of IL-1 to the IL-1 receptor, results in threefold reduction in mortality. It is hoped that these experimental results will lead to clinically useful products for the treatment of bacterial septic shock in humans. Then, bacterial toxic shock is caused by superantigens. A variety of microorganisms produce toxins that act as superantigens. Superantigens bind simultaneously to a class 2 MHC molecules and to the V beta domain of the T cell receptor, activating all T cells bearing a particular V beta domain. Because of their unique binding ability, superantigens can activate large number of T cells irrespective of their antigenic specificity. Although less than 0.01% of T cells respond to the given conventional antigen, between 5% and 25% of T cells can respond to a given superantigen. The large proportion of T cells responsive to a particular superantigen results from a limited number of TCR-V beta genes carried in the germline. Mice, for example, have about 20 VB genes. Assuming that each VB gene is expressed with equal frequency, then each superantigen would be expected to interact with 1 in 20 T cells, or 5% of the total T cell population. A number of bacterial superantigens have been implicated as the causative agent of several diseases such as bacterial toxic shock and food poisoning. Included among these bacterial superantigens are several enterotoxins, exfoliating toxins, and toxic shock syndrome toxin from Staphylococcus aureus, pyrogenic exotoxins from Streptococcus pyogenes, and Mycoplasma arthritis supernatant mass. The large number of T cells activated by these superantigens result in excess production of cytokines. The toxic shock syndrome toxin, for example, has been shown to induce extremely high levels of TNF and IL-1. As in bacterial septic shock, these elevated concentrations of cytokines can induce systemic reactions that include fever, widespread blood clotting, and shock. Cytokine activity is implicated in lymphoid and myeloid cancers. Abnormalities in the production of cytokines or their receptors have been associated with some types of cancer. For example, abnormally high levels of IL-6 are secreted by cardiac myxoma cells, a benign tumor, heart tumor. So, myxoma and plasma cytoma cells, and cervical and bladder cancer cells. In myeloma cells, IL-6 appear to operate in an autocrine manner to stimulate cell proliferation. When monoclonal antibodies to IL-6 are added to in vitro cultures of myeloma cells, their growth is inhibited. In addition, transgenic mice that express high levels of IL-6 have been found to exhibit a massive fatal plasma cell proliferation called plasmacytosis. Although these plasma cells are not malignant, the high rate of plasma cell proliferation possibly contribute to the development of cancer. Then, Chagas disease is caused by a parasite. The protozoan Trypanosoma cruzi is the causative agent of Chagas disease, which is, I'm not sure about the pronunciation, sorry if I am mispronouncing. It is characterized by severe immune suppression. The affinity of this Trypanosoma cruzi to mediate immune suppression can be observed by culturing peripheral blood T cells in the absence and in the presence of T cruzi, and then evaluating their immune reactivity. Antigen, mitogen, or anti-CD3 monoclonal antibody normally can activate peripheral T cells. But in the presence of T cruzi, T cells are not activated by any of these agents. The defects in these lymphocytes has been traced to a dramatic reduction in the expression of the 55 kilodalton alpha subunit of the IL-2 receptor. As noted earlier, the high affinity IL-2 receptor contains alpha, beta, and gamma subunits. The alpha subunit is specific for cytokine binding. So, co-culturing of T cells with T cruzi and subsequent staining with fluorescent labeled anti-TAC, which binds to alpha subunit, revealed a 90% decrease in the level of alpha subunit. Although the mechanism by which T cruzi suppresses expression of the T subunit is still unknown, the suppression can be induced across a filter that prevents contact with the lymphocytes and protozoa. This finding suggests that a diffusible factor mediates clinical applications for regulating the level of activated T cells in leukomas and autoimmune diseases. The next therapeutic uses of cytokines and their receptors. The availability of purified cloned cytokines and soluble cytokine receptors offer the prospect of specific clinical therapies to modulate the immune response. A few cytokines, notably interferons and colony stimulating factors such as GM-CSF, have proven to be therapeutically useful. However, despite the promise of cytokines as powerful mediators of immune and other biological responses, not many have made their way into clinical practice. A number of factors are likely to raise difficulties in adapting cytokines for safe and effective routine medical use. One of these is the need to maintain effective dose levels over a clinically significant period of time. During an immune response, interacting cells produce sufficiently high concentrations of cytokines in the vicinity of target cells, but achieving such local concentrations when cytokines must be administered systematically for clinical treatment is difficult. In addition, cytokines often have a very short half-life, so that continuous administration may be required. For example, recombinant human IL-2 has a half-life of only 7 to 10 minutes when administered intravenously. Finally, cytokines are extremely potent biological response mediators and they can cause unpredictable and undesirable side effects. The side effects from administration of recombinant IL-2, for instance, range from mild, that means fever, chills, diarrhea, and weight gain, to serious such as anemia, thrombocytopenia, tinea, shock, respiratory distress, and coma. Despite these difficulties, the promise of cytokines for clinical medicine is great, and efforts to develop safe and effective cytokine related strategies continue, particularly in areas such as inflammation, cancer therapy, and modification of the immune response during organ transplantation, infectious disease, and allergy. Some specific examples of various approaches being explored include cytokine receptor blockade and the use of cytokine analogs and cytokine toxin conjugates. For instance, proliferation of activated T cells and activation of TC cells can be blocked by anti-TASP, the monoclonal antibody that binds to an alpha subunit of the high affinity IL-2 receptor. Administration of anti-TAC has prolonged the survival of heart transplants in rats. Similar results have been obtained with IL-2 analogs that retain their ability to bind the IL-2 receptor but have lost their biological activity. Such analogs have been produced by site-directed mutagenesis of cloned IL-2 genes. Finally, cytokines conjugated to various toxins, the beta chain of diphtheria toxin, have been shown to diminish injection of kidney and heart transplants in mammals or animals. Such conjugates containing IL-2 selectively bind to and kill activated T cells. Cytokines in hematopoiesis. Early work in Australia and Israel demonstrated that soluble factors could support the growth and differentiation of red and white blood cells. The first of these subtle soluble factors to be characterized, erythropoietin, was isolated from the urine of anemic patients and shown to support the development of red blood cells. Subsequently, many cytokines have been shown to play essential roles in hematopoiesis. During hematopoiesis, cytokines act as developmental signals that direct commitment of progenitor cells into and through particular lineages. As shown, a myeloid progenitor in the presence erythropoietin, antibiotin, a surgical lineage of absence, a failure of genetic microvesical oxidants, superoxide and others on the basis of CCD and some other aspects, would proceed down a pathway that lead to the production of erythrocytes. Suitable concentrations of a group of cytokines including IL-3, GM, CSF, IL-1 and IL-6 will cause it to enter differentiation pathway that lead to the generation of monocytes, neutrophils and other leukocytes of myeloid group. The participation of leukocytes in immune response often results in the death and removal. However, both adaptive and immune response generate cytokines that stimulate and support the production of leukocyte. The steps at what number of cytokines participate in hematopoiesis is very important, which you can see with the help of this table. So cytokines are low molecular weight proteins that are produced and secreted by a variety of cell types. They play major roles in the induction and regulation of cellular interactions involving cells of immune, inflammatory, and hematopoietic systems. The biological activities of cytokines exhibit pleiotropy, redundancy, synergy, antagonism, and in some instances cascade induction. There are over 200 different cytokines, most of which fall into one of the following families: hematopoietins, interferons, cytokines, and tumor necrosis factors. Cytokines act by binding to cytokine receptors, most of which can be classified as immunoglobulin superfamily receptors, class 1 cytokine receptors, class 2 cytokine receptors, members of TNF receptor family, and chemokine receptors. A chemokine can only act on a cell that expresses a receptor for it. The activity of particular cytokine is directed to specific cells by regulation of the cell's profile of cytokine receptors. Cytokine-induced multimerization of class 1 and class 2 cytokine receptors activated JAK-STAT signal transduction pathway. Antigen stimulation of Th cells in the presence of certain cytokines can lead to the regeneration of subpopulation of helper T cells known as Th1 and Th2. Each subset displays characteristic and different profiles of cytokine secretion. The cytokine profile of Th1 cells support immune responses that involve the marshalling of phagocytes. CTLs and NK cells also to eliminate intracellular pathogens. Th2 cells produce cytokines that support production of particular immunoglobulin isotype and IgE mediated response. Therapies based on cytokines and cytokine receptors have entered clinical practice.